Nervous system. That's what we're finishing out the semester with. We're going to talk about the central nervous system, the brain and the spinal cord. And then the last chapter we're going to discuss is about the peripheral nervous system as it relates to how your body subconsciously controls all of your bodily functions. Now, I just really thought that was a cool picture. You know, when you look at biology and you look at the human body, I know that us nerdy scientists, we love to say, oh, that's beautiful how this works, the integration and homeostasis. But sometimes when you just put things together, you just have to really admire the beauty of the human body and the connections that you see simply within the human brain. So that's what we're focusing on today. The central nervous system, and today, and probably through the end of the week, we're going to cover the brain, all the parts of the brain stem, cerebrum, cerebellum. And then next week, in Dead Week, we will get into the peripheral nervous system. So, when we look at the brain, this thing that resides inside of our skull, you probably, again, have seen this in lab, that the brain is going to be comprised of dark and light tissue. And the dark tissue that you see on the periphery is referred to as the gray matter. And the material on the inside that's lighter in color, that's the white matter. Start with white matter. What gives white matter its white color? Myelination. What are the only processes of neurons that are myelinated? Maybe that's kind of a tricky question. What are the only processes of a neuron that are myelinated? The axons. Dendrites are not myelinated. Why are dendrites not myelinated? Okay, let's do this. What is myelin? What's it there for? Speed up what? An action potential. So why aren't dendrites myelinated? You can't have an action potential in a dendrite because what channel is missing? Sodium voltage gated channels are missing. So there's no need to put myelin around dendrites. So where you see white matter, you know it's myelinated, but it's myelinated axons. So if it's not white, that means myelin isn't present. Then we're talking about the other cells and the other cell processes that are unmyelinated. So now we're talking about cell bodies. We're talking about dendrites. We're talking about neuroglial cells. All of those cells are present in the gray matter. So typically that's the cellular part, whereas the white matter are the tracks and the passageways through which signals are being sent. Bless you. All right, so we said the brain sits within our skull, aka the cranium. And so it's all of these flat bones that are fused together into this thing we know as the cranium or the skull or the brain case. Now, you know there's multiple bones in the skull, right? You did that in the skeletal system. And when babies are born, those plates are not fused together. Because have you ever seen a baby that was born through a natural vaginal delivery? And maybe the mom's been in labor for a considerable amount of time. What's the shape of that baby's head? Cone head. That's because the plates of the cranium slid over each other so that it would make it a little bit easier for this melon to get out. It's easy for guys to say, right? If the bones were fused together, the head could not change its shape. It would be much more difficult to deliver the child vaginally. And so because of the spacing and because those plates haven't fused yet, haven't grown to their full capacity yet, you have these spots. The soft spot, have you ever seen the soft spot in a baby's head? Okay, you can actually put your finger there and feel a pulse in that soft spot. Did you learn the big fancy name for that in lab? Starts with an F, called the fontanelles. That's the main fontanelle that's present we typically see as the soft spot. Well, beneath these flat bones of the skull, we're going to have protective membranes that collectively are called the meninges. And in a health setting, you may actually encounter an infection of the meninges, and what do you call that disease? Meningitis. Do you see where they get that name from? It's an infection and inflammation of the meninges. Now, as we're going down, we go through the epidermis of our skin, the scalp. We go through the bone, the flat bones of the skull. Then we encounter the tough outer layer, which is called the dura mater. Now, this is going to be somewhat continuous with the periosteum of those flat bones of the skull. But if you are in lab, if you've got a fresh specimen of a brain, and I think they use sheep brains in lab, some of those may have this white, sort of pearlistic looking membrane around them. Did anybody's brain have that membrane around it? Were you able to just tease it apart, peel it apart? No, you got to cut it because that stuff is tough. That was the dura mater, this very tough envelope that surrounds the brain. Now, once you penetrate through the dura, then you end up in a layer that is called the arachnoid. Now, arachnoid sounds like what? Arachnid, spider. Because this arachnoid layer, it has an arachnoid matter, a membrane. It actually has two of them. And in the middle, it has these trabeculae. Let me just go ahead and use that word. When we say trabeculae, we usually think of what tissue? Bone, connective tissue. But in this case, it has these interconnected girders of tissue that extend between the arachnoid matter on the outside and the arachnoid matter on the inside, creating this very sort of a spacious, spongy area that is called the arachnoid space. So think of it like a big I-beam running through just underneath the dura mater. And all of this space is going to be nice because it's filled with cerebrospinal fluid. That's CSF, cerebrospinal fluid. And you have a number of blood vessels that run through this arachnoid layer. We'll see that in the illustration coming up next. Now, as you go through the dura, you go through the arachnoid, you finally get to the last layer, the innermost layer before you get to the brain tissue, and that is called the pia mater. Now, it says this is a delicate inner layer. The pia is actually adhered to the surface of the cerebral cortex. In the lab, you cannot peel the pia mater off the brain without removing brain tissue itself. That's partly how delicate it is, it's partly how thin it is, and it's partly how closely adhered to the brain the pia mater actually is. So we've got the cranium, the three meninges. We've talked about the cerebrospinal fluid, that hydraulic cushion that's going to protect us against shocks, mechanical shocks. But remember, because it's flowing, it also helps to create kind of a protective moat against infection. So that's another function of the cerebrospinal fluid. And while we can think of the cerebrospinal fluid as contributing to the blood-brain barrier, what glial cell do we attribute the blood-brain barrier to? Astrocytes. Those are the ones that really provide the fully functioning blood-brain barrier. But we're going to go ahead and say the cerebrospinal fluid flowing around helps and contributes to that as well. So here's our illustration. There we have our skin, the scalp. Here we have our flat bone. This is the periosteum on the outside, just sort of in contact with the dermis of our skin. Here we have the periosteum on the inside of the bone, and that's going to be continuous with our dura. They're saying that's going to be our dura. Do you see this spider web looking stuff here? That's the arachnoid. See kind of why they named it that? This arachnoid layer's got a lot of space, a great space for our cerebral spinal fluid to flow, but then also we have our blood vessels penetrating through the arachnoid space. Then the very thin kind of purplish line, is that purple, I guess? And you can see it extending into the folds in our cerebral cortex, that is the pia mater. And those are our 3 protective meninges. And again, primarily these meninges are there to prevent infection, to maintain the inner area for our cerebrospinal fluid. Now, these meninges aren't just for the brain. They're going to extend down into the spinal cord as well. Did you know that the meninges went into the spinal cord? So we've got our brain, this control center of the central nervous system. It's protected inside this bony case. It's wrapped in these membranes. It's got this fluid that flows around. But it's also got these cavities that are interconnected on the inside of the brain, and you can see that flows through these passageways down into even our spinal cord. And so these cavities are called ventricles. I think we mentioned them last time we talked. Ventricles have nothing to do with the heart. They're just cavities, empty spaces. And so we have these ventricles that are sort of in the base of the cerebrum. We have a deeper 3rd ventricle. The 4th ventricle is getting into the brainstem, and that leads into our central canal of the spinal cord that we'll see throughout the spinal cord. And it's within these cavities is really one of the places, in addition to around the brain and around the spinal cord, where we have a flow of cerebrospinal fluid. And again, the protection, hydraulic, any sort of mechanical vibration, we're going to be protected from that. We're also sort of immunologically protected. We're washing physically pathogens away from the actual central nervous system tissue. And so these are all important functions of maintaining the proper health and integrity of the central nervous system. Because even in lab, even though you've got preserved tissue, the brain's delicate. It's extremely delicate. Yours is preserved, and it's going to feel tougher than a natural, freshly dissected brain would. But even the one you have in lab, I think you're very aware the brain's a very delicate organ. All right, so now let's start to tear apart our brain. And our brain is really going to be comprised of what we're going to talk about 4 major control centers. You're pretty familiar with the cerebrum and the cerebellum. We have this deeper part of the brain that has this really weird sounding name to it, the diencephalon. We're going to get to the crux of why we call this the diencephalon in the next couple of slides. But then we finish off at the top of our spinal cord, we have this thing called the brain stem. And you can see we've colored these here. The diencephalon is in purple. We got the brain stem in green, which is composed of the midbrain at the top. This enlarged pons is the second portion. And then the portion, again, it's hard to describe where does the spinal cord start and where does the brain stem stop and all that. But at the very top of the spinal cord, we have the medulla. Now, I've explained this to a few students, so I'll just go ahead and explain it to the class. You will only ever hear me refer to this thing as the medulla. I cannot say both words, because when I say both words, I hear Colonel Sanders on Waterboy, and I start to laugh. You know what I'm talking about? You've never seen Waterboy? Yes, you have. Are you serious? Waterboy? Waterboy? You know, this big biology professor at his university looks like Colonel Sanders, and he always says the medulla oblongata. I mean, Mama said, I can't go there. So, medulla. I say medulla, you think whatever you want to think, but it's got two names. I can't do it. All right, the other thing, we're going to spend a little bit of time, but we have 12 cranial nerves that leave the brain and go out to different parts of the body. One of these cranial nerve, number 10, is called the vagus nerve. Vagus in Latin means wanderer because the vagus nerve goes all over the body. It's controlling heart, it's controlling respiration, and we'll see more about where these go and what they do when we get into our last chapter on the autonomic nervous system. But just know there's 12 of them. I briefly want to touch on the development of our nervous system, just very quickly, because this is where that term diencephalon comes from. So we're not going to spend a lot of time on this, even though as a developmental biologist, I really, really want to, because I love this stuff. But one of your earliest baby pictures, if you could do an ultrasound when you were probably, I don't know, about 7 days post-conception, your earliest baby picture is going to look like a stack of 3 pancakes. These oval-shaped pancakes, 3 of them, that's you. And your central nervous system is going to begin as that top layer starts to roll up like a jelly roll. You see how it's flat here and rolling up in the middle? That's the same tissue that's going to form your skin, but right down the middle, those cells change and they become your neural plate. And as that neural plate starts to roll up, and you can see in these lower illustrations, you see how it's rolling up and it's going to fuse together at the top. It pulls that lateral tissue toward the middle, and that lateral tissue fuses into the skin all along your back. And that part that rolled up now is more inferior. It's deeper to that tissue, and that's going to be your neural tube, and that's going to develop into your central nervous system, all along the length of your body. All right, we're good with neural tube? All right, so now this picture, you start on the left. That's just simply an early picture of your neural tube. And from that, all parts of our central nervous system develop. The bottom part, that's going to be our spinal cord. But we first are going to break down this region at the top into forebrain, midbrain, and hindbrain. Fore is in front, hind is in back, and mid, of course, is in the middle. But notice the names that we get from these. The forebrain are prosencephalon. Pro means in front, before. The prosencephalon further differentiates into the telencephalon, which is in the front or at the top. That's your cerebrum. The diencephalon is the next version down. It's still part of the forebrain from earlier in embryonic development. The diencephalon is the epithalamus, thalamus, and the hypothalamus. We're going to describe those as we get into the diencephalon portion, so don't freak out yet. Okay? Midbrain is mesencephalon because mes means middle. Then the hindbrain, the ramencephalon, is going to be the met, which is the cerebellum and the pons. And the myelencephalon, like myelin, part of the spinal cord, that's going to be the medulla. So do you see, this is simply a breakdown. Encephalon means brain, because if you have an infection of the brain, what disease? Encephalitis. So this is where we get that term diencephalon for one of those portions. That's going to be the deeper portion of the brain where we're going to have these structures. So as far as the divisions of the brain, the only one of these names that I'm going to expect you to remember is the diencephalon. We're going to have all these others associated with it, but it really doesn't concern me that you know these with each one. But the diencephalon is the only one we have to go with it. Okay? Make sense? Did you have a question?
Yeah. So for spina bifida, does it have to do with the neural tube?
So the question is spina bifida. Does it have to do with the neural tube? And the answer is absolutely yes. You can see in the illustration here in the middle, we have had fusion of these two, what are called our neural crests and neural folds that have fused together in the middle. Along the length of this neural tube, you're going to have several points that start zipping up. And if during your embryonic development it doesn't completely zip up, that's your spina bifida. It usually happens lower down in this lumbar region. It can happen anywhere, but it usually happens down here. Now, you're not going to see a picture. I wouldn't do this to you. It's in your book, actually, if you wanted to look it up. But you have to zip up at the top and you have to zip up at the bottom as well. And if you don't zip up at the top, you do not form a cerebrum, and most of the time you do not form a cranium. The condition is called anencephaly and acrania, but the babies are delivered at term. They have a heartbeat. They breathe because the brain stem that controls that developed, but they have no higher brain function. And so it's not compatible with life, and they only live a very short time after delivery. Well, encephalopathy, I mean, that's going to be a pathology of the brain. And, anencephaly, an meaning no cephalon, no brain. I mean, that's the pinnacle of that pathology.
I'm sorry, did you say the neural plate becomes the neural tube?
Yeah, so the neural plate is just this flat, you see how this purple and kind of blue color?
Yes.
It's just going to be this sort of medial portion of this top layer called the ectoderm. That's going to turn into the neural tube.
Okay, thank you.
Yeah. All right. So we're good with the diencephalon. I didn't want to throw out a term and go, well, where the heck did that come from? Dr. Dye, name it, yeah. So anyway. So we're going to start, again, the telencephalon, our cerebrum. We're going to spend most of the rest of our time today talking about this and probably on Friday. Our cerebrum, all of our reasoning, all of our thought, all of our memory is housed in the cerebrum. Voluntary motor activities. You willing yourself to do something begins in the cerebrum. Walking, running, talking. All of our conscious sensory activities, visual, auditory, olfactory. There's a word that is often used in the scientific community you may not be familiar with, gustatory. What is gustatory?
Taste.
Smell. Gustatory is smell. No, I'm sorry, you're right. Olfactory is smell, gustatory is taste. I'm sorry. Gustatory is taste. Now, when you look at the cerebrum, and we're going to be talking about adult organs, we see we have a right and left hemisphere, two halves of the brain, that are divided by a longitudinal fissure that runs down through the middle in a dorsal ventral direction. Now, these two halves are not completely separate. You do have cables, interconnecting axons that go back and forth between right and left sides. When we get to the deeper parts of the brain, we're going to see that commissure, which is a connection between halves, and the major commissure of our brain is called the corpus callosum. That's the interconnecting pathways, but the actual hemispheres are separate from each other. So as we mentioned before, we have the gray matter, the white matter. The gray matter on the outside is going to form what is called the cerebral cortex. Now, do you remember those terms? We've not talked about them exclusively, but cortex versus medulla. One is on the outside, one is on the inside, or in the middle. That's how I remember medulla middle, because they both start with M, which means cortex is on the outside. Now, anytime we need to have more of something to accomplish a task, but we only have a limited amount of room, think of our intestines and their ability to absorb material. How can we absorb more material in our intestines in a defined space? Increase the what? Surface area. So if we need more surface area to house all of our neuronal cell bodies and dendrites, but we got to fit it inside the skull, how do we increase that? Increase the surface area of the cortex, and that's why we have all the folds and the grooves. The folds are a gyrus, gyri plural. The grooves is a sulcus or sulci. That's the plural. But that's why it's folded like that, to increase the surface area so we can pack in more neuron cell bodies. So the gray matter cortex on the outside, all of our myelinated axons on the inside, that's where we have the white matter. And even in this illustration, you see this bridge going from right to left right there? That commissure, pardon me, that commissure, and the main one we have is the corpus callosum. And that's its purpose, to interconnect both sides. Now, we also have within our brain, in these deeper portions surrounded by the white matter, we have these other gray areas, and these are called cerebral or basal nuclei. Now, do you remember how we define a nuclei in the central nervous system? It's a ganglion in the peripheral nervous system, but we call it nuclei in the central. And what is, by definition, what is a nuclei in the central nervous system? A collection of nerve cell bodies. And if it's a collection of nerve cell bodies, that's why it's gray matter. It's unmyelinated. And you can see several of these basal nuclei present surrounded by our white matter. Now, these aren't really a major functioning aspect as far as the regulation of movement and the maintenance of tone. We're going to see the cerebellum plays a much more critical role in that. But so far as we understand it, they do assist in motor movement and muscle tone. And so we'll ascribe that function to these, as we'll just leave them right now, our generic basal nuclei. All right, so looking at the cerebrum from a gross anatomical aspect on the outside, we can divide this up into 4 visible lobes. This one's easy. What's this one? Frontal. It's in the front. This next one is kind of easy too, on the sides. Temporal. Because man, if you've got a headache right here, what's that location? What do we call that? Our temples. That's why we call this our temples, because that's over the temporal lobe. Then sort of at the top toward the back, we have the parietal lobe, and the very back, we have the occipital lobe. And again, we're looking at one hemisphere, but understand this is bilaterally symmetrical, so it's the same on both sides. All right? Now, there are 2 lobes that we can't envision just from the outside in the gross anatomical view. One that's deep to the lateral sulcus, and you see we've removed the lateral sulcus. This is our temporal lobe. So it's deep into this lateral part of our frontal and parietal lobes. That's called the insular lobe. And then as we get deep into the brain, in this area of the corpus callosum, and sort of making an arching around in the same pattern as the corpus callosum, we have what's referred to as the limbic lobe. This limbic lobe is going to be part of this connection with the primitive part of our brain, where we have behavior and aggression. Sleep patterns are also controlled here in this limbic lobe. So 6 lobes overall, the 4 we see and the 2 we don't, the insular and the limbic. Now, what does it do? What is the cerebrum good for? Well, certainly motor function. We've already said that. Our willing ourselves to accomplish some task and work begins in the cerebrum. And it begins in the area where we see this sort of blue band. And we can see this is going to be around what is called the precentral gyrus. It's almost right in the middle. And what they have done here in this primary motor cortex, they have taken a coronal slice. This is going to be our longitudinal sulcus here along the right-hand side. And what they've done is they have mapped out the different places of the body where the initiation of movement is controlled. So your knee and your hip, that's almost at the very superior part of our frontal lobe in this primary motor cortex. And then as we move on around and get lower, we're going to see that we finally have the neck and the head and the face and the lips, then we get into the pharynx and the tongue. This sort of mapping out of the human body onto the brain is referred to as a homunculus. Now, this is kind of a jacked up homunculus, because we've got the feet down here, we've got the hands reaching up here, and then the head, which we kind of expect to be here, is on down the line. But anyway, a homunculus is a representation of the human body onto our primary motor cortex. Now we also have motor, we will ourselves to speak. And so our motor speech area, also called the Broca area, this is going to be our left frontal lobe, and they've illustrated it here a little bit in this blue. But this is going to control our speech. So that's why, depending on the type of head trauma you may have had, I mean, if you take a really good collision and your head hits a windshield, or in a football game, if you take a hit in the right place and you get a concussion, if it happens to affect that left area, you know, people start slurring their speech and you really can't understand what they're saying. It's because that's affected that motor speech area in the left hemisphere and the left frontal lobe. Now, like we had a primary motor, we've got a primary somatosensory cortex. And again, this is going to be a little bit more dorsal, if you will, from our primary motor, which is just in front. They're very close to each other. And so we're going to be receiving a lot of information. Again, you see our homunculus. Feet, we have our genitals are the deepest ones to the brain, but we've got our feet, our hips, our torso, then our arms and hands. Then we have our face, our mouth, and sort of our tongue, pharynx, and the abdominal regions, the homunculus drawn out. Now, this is on both sides. You don't just have one on one side and the primary on the other. You see how it's on both hemispheres. So what kind of sensory information are we getting? Well, certainly we get touch and pressure receptors. We've got thermoreceptors. Are we hot or cold? Pain receptors, which are called nociceptors. You may not have heard that terminology before. Nociception is pain. But our other one that's extremely important is proprioception. Information is coming to our brain about the contraction state of our muscle, the tension that we have on our joints. It gives us an idea of the position of our body and our appendages in space, as well as are we stopped, are we accelerating, that kind of information. Because without that information, that instant and immediate feedback, walking would be impossible.